Welcome to Study Loop.
Hello students, the question says that you are determining the structure of a new protein when you perform molecular exclusion chromatography. You determine that the size of the protein is 300 kDa kilodalton. Next, you perform an STS page and observe one protein band at 150 kilodalton. After running the gel, you realize that you forgot to add beta-mercaptoethanol to the protein mixture before running the gel. So you repeat the analysis. The second time you run an SDS page, this time with the beta-mercaptoethanol, there are two protein bands which correspond to 100 kilodalton and 50 kilodalton. Now, on the basis of this information, we are being asked certain questions. First one is that given what you know about the levels of the protein structure, protein purification, and protein detection techniques, what is the function of SDS in the gel? We have been given options, and the correct options are B, C, and D. And option B says that SDS gives the protein negative charge. And option C is SDS disrupts disulfide bonds. And option D is SDS disrupts weak interactions or tertiary and quaternary structure. Another question asked is, what is the function of beta mercaptoethanol? And the correct options are option A and option B. And the option A is disrupts weak interaction needed for tertiary and quaternary structure. And option B is disrupts disulfide bonds. Another question asked is, does this protein have a quaternary structure? Yes or no question is this. As we know that during the SDS page, we got two protein bands, which means that it contains different protein domains which interact with each other to form a fully functional protein. That means it has a quaternary structure. Yes, this statement is true because in quaternary structure, different domains come and they interact with each other and form a functional protein. Another question asked is, how many subunits does this protein have? So as we know that we got the bands 100 kb and 150 kilodalton. So which means that it has two subunits. One is 100 kilodalton and another is 50 kilodalton. So the correct option is option C, which says that 3. And another question asked is, does this protein have domains? Yes or yes not? Yes or not, it is a yes no question. Yes, the protein has domain, that's why they interact with each other, form a quaternary structure. Sorry, not subunit, but is asking for the does this protein have domains, which means that domains are specific protein sequences which form a different structure, but by the SDS simply and SDS page we cannot determine the different domains in a protein. We need a further analysis to if determine the presence or absence of a protein or domains in a protein, or how many domains are present in protein. So the correct option is option C, which says that cannot determine from information given. I hope you found the video useful. Thank you.